It's 9 o'clock here in San Diego, so we'll begin the webinar. Today's webinar is on ligand design using the ICM interactive ligand editor. My name is Andrew Ori, and if you have any questions after the webinar, please feel free to email me or call. During the webinar, you can use the questions panel to ask questions, and I'll try to answer them during the webinar or at the end. If you want to see a recording of this webinar, you can find it on YouTube, and also keep up to date with future webinars on social media. A 30-day ICM Pro license is available for all webinar participants. As I mentioned, you can catch up with the other webinars in this series, from ligand docking to virtual screening to induced fit, flexible docking and screening, covalent docking, and fragment design on YouTube here. So today's topic is about the ligand editor. So the editor is widely used throughout the pharmaceutical and biotech industry for lead design of your ligand and optimization. So usually you would use it in a situation where you have a lead chemical, and ideally you have a high resolution crystal structure with the ligand bound, and then you can use that structure to optimize the ligand. Or alternatively, you can dock and you can predict the interactions beforehand without a crystal structure. So, but in an ideal situation, you'd have a crystal structure with the lead compound, and then you can start modifying the compound. So you can modify the chemical in either 3D, so you just click on an atom and it will change or delete, or you can modify it in 2D in this left-hand panel. And you'll immediately see the effect of the change on the docking score, and this will give you an indication of whether the modification is good or bad, or you can monitor the changes here. So this was developed in close collaboration with medicinal chemists at Novartis, and they developed a product called Focus, which they use globally around in Novartis, and part of this focus was the development of this editor, as well as a lot of other cheminformatics tools for medicinal chemists. So just as an example of a recent success story using the editor, back in August this year, the FDA granted approval of Vabomere to Rempex, which is part of the medicines company, and it was part of it was the structure-based design of the molecule was developed using the ligand editor and other tools in ICM. So it's a novel cyclic boronate beta-lactamase inhibitor, and the structure-based design was led by Max Totchoff here at Molsoft using ICM Pro, which contains the ligand editor. The novel cyclic boronate chemotype was first modeled in silico and prioritized for synthesis based on modeling and docking into several beta-lactamase target enzymes. And there's a paper describing this work here. So today's topics are how to use the ligand editor to visualize the interactions between your ligand and the receptor, how to use the editor to make changes in 2D to your ligand or in 3D, how to evaluate the effect of a modification on binding, how to undo and redo the changes that you made, and also to save changes that you like to a spreadsheet, how to then perform ligand docking or minimization between the ligand and the receptor, and how to refine that, how to use distance restraints and tethers during docking, and how to do substituent virtual screening on your molecule. We've already showed how to do covalent docking in the ligand editor, as well as fragment docking and methods to incorporate induced fit, such as multiple receptor conformation 4D docking, and you can view those on YouTube. And then next week we'll go into more detail about how to use it for ligand design using atomic property fields. That's the webinar, the final webinar in the series next week. So in this example, we're going to go through an example. In this example is we have a lead compound called rosiscovitine. It's an experimental drug candidate for CDK. It preferentially inhibits enzyme targets including CDK2, 7, and 9. It alters the growth phase or state within the cell cycle of treated cells. And it's in clinical trials at the moment for Cushing's disease, leukemia, and other infectious such as HIV and cystic fibrosis. So we're going to look at this structure. We're going to look at how the drug interacts with the receptor by displaying receptor property surfaces. Look at the hydrogen bonds that are made between the ligand and receptor. How to display what we call energy circles. This enables you to keep an eye on the good interactions between your ligand and the receptor, and those such as these red marks around this one, for example, that are bad. They're making a clash with the protein. And you can also display things like unsatisfied hydrogen bonds and other interactions as well. So let's just go to ICM, and I'm just going to open that. You should have an icon here with ICM, and it will open the graphical user interface. Now the protein PDB code for the protein we're interested in is 1UNL. So we go to the search tab, type in 1UNL and press search. When we open it, we can see that we have 4 protein chains, yellow, orange, blue and green. Only one of them actually only has the ligand bound, so it's this riscovitine here that has the ligand bound. So I'm just going to delete the other, just for clarity, I'm just going to delete the other chains. So right click and delete. And then we can just center on the ligand, just have a look at the ligand. If you want to see the interactions of the ligand, we can go to right click and say ligand pocket interactions, and we can see that it's a kinase inhibitor. We have the N terminal here with the glycine-rich loop, the hinge region, and the C terminal here. Okay, so in order to edit this ligand and try and explore different ways of improving it, we can use the ligand editor. To use the ligand editor, we go to Lig Edit tab, shown here, and we first need to tell ICM which is the ligand that we want to optimize, and that is riscovitine here. So we just click on, we go to Lig Edit, we click on the first button along, which is the Setup Ligand, and it asks us which is the ligand molecule. Sometimes you may have more than one chemical in the PDB, and so you'd have to use the drop down to find it, but we only have one, so that's just, it automatically finds it. And the you can also assign the charge based on our pKa model here. So we just go OK. You can see that now that ICM has separated the ligand out, we have this new object called 1UNL underscore lig. And so this is the ligand that we will actually modify. We can, and you can see it has this little symbol here, which means that this is the ligand that's going to be modified. So now we need to tell ICM which is the receptor. So we go to the next button along, and we use the protein tab, which is open by default. This APF one is for ligand-based approach, but we're going to use a protein. The receptor object is 1UNL. It automatically finds it because it's the only protein chain that we have in ICM. Then you have the option to keep tight waters, and that would be 3 or 4 hydrogen bonds between the ligand, the water, or the receptor. Or you can say keep all waters. So if you had a single water that you know that's important, that probably not going to be displaced by the ligand, you can just delete the other waters and keep it here and say keep all waters. Or alternatively, just say delete all waters. And now we're converting an X-ray structure into an ICM object, and so we're adding hydrogens to the structure. So we need to optimize hydrogens. We need to optimize the positions of certain residues where the crystallographer may not necessarily have seen the correct orientation of that side chain. So things like asparaginins and glutamines and the histidines to optimize the hydrogen bonding network. And if there are some atoms on a residue which have not been seen in the density and have a zero occupancy, then ICM will mask those out and won't explicitly model them. If you want ICM to model those missing atoms, you just uncheck here. Likewise, if you prepared your protein structure in a different program and you already optimized all the hydrogens and you're happy with the structure, then you can just uncheck everything here and go ahead. So now we have two options. We just need to tell ICM where the pocket is, basically where we're going to dock. So we could run ICM Pocket Finder by clicking on this button here, and you see in this structure we find an allosteric pocket here in the C terminal, but this one here is the ATP binding pocket. Or in most cases, when you're using the ligand editor, you already have the ligand bound, so you can just say make box around existing ligand, and this box is where we generate the maps for docking and for optimization. So if you're building, in many cases with the ligand editor, you're going to be building out from your original molecule. So you need to just double check that the box encompasses the region where you are building out to. I may want to, in this example, we're looking down on top of the N-terminal region, and this box is not actually taken into account the tip of this glycine-rich loop. So you may want to just increase it a little bit, and you drag it using the corners of your cursor key and just drag like that. Okay. And now ICM will make the maps. It takes a little bit of time, so I'm just going to close it, and I've got an example ready-made. So just open ICM. It takes a couple of minutes to make the maps. So once it's finished, you have something like this. So we have the ligand. We can see again the hydrogen bonds with the hinge. There's another hydrogen bond here. So now we are ready to look at the interactions between the receptor and the ligand. So most of the visualization tools are on the right hand side, these ones here. This one will center on the ligand. Just a simple click on there. And I also should point out that now we set up the receptor, we also have an additional object called 1UNL_rec, which means it's the receptor. Likewise with the ligand, we had this little icon. This now has this, you may not see it very well in GoToMeeting, but it has a small green, supposed to be represent a receptor. So now that's the receptor. This is our original PDB file that we were using. And these are the maps that we will dock to. You can electrostatic interaction, grid potentials, and other van der Waals, hydrogen bonding, and things like that. Okay, so that's the center. You can see the hydrogen bonds are displayed. We can toggle those on and off using this button here. We can also just change the view. So there's inbuilt, we have what we call pretty view, which you can modify to your own taste. But if you click on that, we get this display here. I'm just going to remove the spheres, and we can see the interaction between. It's a little bit easier to see. And also for this example, I'm just going to color the ligand a different color just to make it clearer which is the ligand. So I'm just going to double click on the ligand, go to Display tab. I see that the ligand is displayed in X stick, this thicker stick. So we can click and hold and say color C only, color the carbons only, and choose green, some other color. Okay, so this is the ligand we're going to modify. But it'll be also, before we start modifying, it would be a good idea to look at some other interactions. So if we go back to the Ligand Edit tab, we can click on this button here, and then we can choose some other options. So the first one we'd look at is the receptor pocket. So this is your surface of the binding pocket, colored by property, so a binding property. So green is hydrophobic, and blue is hydrogen bond donor, red is hydrogen bond acceptor. So the surface can give you a guide of where you may want to start building or building towards. You can also, there's another option called, if you click and hold on here, you can say display ligand pocket surface, which is a slightly different, it's kind of the opposite of the receptor pocket minus some density. So this actually is quite useful. It highlights, it might be hard to see through GoToMeeting, but it highlights where there's cavities in which to build. So you can see we have a gap here, but if we turn the protein ligand around, we can see that there's no way you can start building from this region here because it's all cut off. There's no density or no space to build into. Likewise, on the other side of the protein, we have a cavity here and a hydrophobic region here that you can maybe utilize to improve activity. Okay, so that's what we call the ligand pocket surface. You can display unsatisfied hydrogen bonds, which will flag regions in the receptor where you could design a hydrogen bond towards. It will show it as a sphere. You can display what we call the relaxed ligand, which is quite important when we start modifying. So when I clicked here to display a relaxed ligand, the relaxed ligand is shown in yellow. So this is the conformation, the energetically favorable conformation of the ligand outside of the pocket. So when you start making modifications, you want to keep an eye on this relaxed ligand. If you deviate too far away, you maybe start seeing to generate your own sort of artifacts in the ligand. Because the ligand is so strained, you're forcing something to make an interaction. So you need to keep an eye on that. But you notice that when we start making modifications, we also have what we call ligand strain. So if this number starts to creep up, you can be sure that your ligand is probably deviating a lot from the original relaxed version of the ligand. So there's those two parameters. Okay, and you may also want to look at other interactions. So for example, we're going to display, you could display the where your polar hydrogens are on your ligand. You just click here. Or you can say I want to see all the receptor polar hydrogens and all the hydrogens on the ligand. You can click and drag here, change different displays, or all hydrogens in the ligand and the receptor. For when we come to start modifying, we have to modify from a hydrogen, so it's sometimes best just to keep ligand all and receptor, this one here, ligand hydrogens basically. We can look at the variables, so the torsion angles are displayed as these ring. Let's zoom in a little bit more. So you can see around the bond there are these rings. Green is good and red is bad. It means the torsion is strained. It's red, but in this case they're all green. And when I show you how to modify torsions, you can keep an eye on this coloring. So that's displayed here. You can change the label style to different, you can show the energy or whatever. Okay. So we also have a button here which shows the what we call atomic energy circles. So if you click on here, green numbers, these are the numbers that contribute towards the score. And so green is good, red would be positive. If you had a clash, you'll see that red highlighting as well as the number will be high. So that's a good way of monitoring your ligand. Okay. So we may also be interested in the direct interactions. What are the residues making contact with our ligand? So we can do that by just clicking on this button here again and choosing Analyze Contacts. And it asks you a question about what if you want to scale the side chain stick thickness based on the contact. So it just highlights those residues making strong contacts are going to be thick, and those that are making very minimal contacts are going to be thin. It's a good guide. 100 will be fine. Just leave it as default and just go OK. And you get this new, this takes a little time, sorry. And you get a table here with the residues that make the interactions. So you have a contact area, so this isoleucine 10. If we double click here and zoom in on the selection, this is a very thick stick, so it's making the biggest contact. And we can see other, so this residue in the top of the pocket is also making a big contact. It's probably loose, yeah, leucine 133 at the top. Okay. And then others that are making small contacts will be a thinner stick, such as this lysine 3089. Should be, yeah, it's down here. It's only making 11 angstroms. So it has also has the distance to the ligand, this residue, the distance to the ligand, and the contact area, and the percent buried. Okay. So you may also, like with the editing, we have 3D option. There's also a 2D option as well. So to view the interactions in 2D, we click on here and go OK. These are some parameters you can play around with to get your 2D diagram, and it takes 30 seconds I think to run, but so while it's running we'll just have a look at the what it will mean. So we will get a little diagram similar to this, so this is a different ligand. Green shading represents a hydrophobic region. Blue shading represents hydrogen bond acceptor. White dash arrows represent hydrogen bonds. As I mentioned, this is a different receptor, so these are different hydrogen bonds. So we've actually got 1, 2, 3, 4, 5, 6 hydrogen bonds in this ligand. Great, when you see grey parabolas, it represents a surface accessible area. So you could build into that region. Broken thick lines, so here we have a thick line, now it's broken, indicates accessible surface area, so we have accessible surface area here. Size of the residue label represents the strength of the contact, so this tryptophan is making a big contact. And in some cases the 2D distance between the residue label, so the distance here between here and here sort of represents proximity, so the ones that are closer are closer in 3D as well. So these ones are a bit further out. Same with this leucine. Okay, so just going to close this. So now you should get a table here with your map of your 2D interaction map, basically. So that's just clicking on this button here. And you can save this as an image. If you right click on it and save as image. You can also modify the ligand as well, rotate I think, and other things, but I'm not going to get into that today. So now we've inspected all the interactions, we can have a better idea of where we want to modify. So I'm just going to open up here. So in 3D ligand editing, you'd have your ligand as we have here in our example, and you can use the panel, which I'll show you in a bit, to how to change. So you just click on the atom here to change the atom, and then click on the atom in 3D. That would change the atom. Same with the bond. Click on the bond type, change from single to double bond, click on a bond, and it would change. Erase will just delete some atoms. You can change the torsions. You can also do virtual screening using a selection of groups as well. And in editing in 2D, you can open this panel. You get a typical editor similar to ChemDraw. You have templates, you have a property monitor, but you can make the changes in here and then commit them to 3D, and it'll give you the score. And everything has full undo and redo capabilities, as we'll see. Right, so let's start editing this. It's going to delete this. I don't need it. Delete. Okay. So to start editing your compound, your chemical, you can do the following. We need to get the Edit Tools panel, so we click on this button here, and we get this panel which I showed earlier. So to change so first of all we need to keep an eye on the score. Sometimes it's a good idea before you start editing, is just to minimize the ligand. So you can minimize the ligand by single clicking, and this will minimize it in the grids, in the energy grids. Or you can minimize, click and hold, and minimize in Receptor, so it will use the side chains of the Receptor to minimize. Okay, so once we minimize, we can then start modification. So for example, to change an atom, we can click on the atom here and just click here, and that would change this from a carbon to an oxygen. You can evaluate the score again. The score actually went higher, and so it's probably not a very good modification. So the lower the VLS score, the better, and you can undo the change and go back to your original molecule with that hydrogen bond. You can change a bond type, so we can click here and change this to a double bond, and then reevaluate the score. It's gone higher a little bit, so it's not particularly good, so we can always undo it and go back to the original molecule. So you can explore different atoms and bond types using these buttons. You may want to play around with the torsion angles of your ligand. So to do that, we click on the this torsion button, and you can see when I highlight over hopefully you can see, let me just zoom in. When you highlight over a bond, you can see my cursor is the cursor key for rotation, but then when I highlight over the bond in order to rotate the torsion, I see the torsion angle here. So now if I use the left mouse button, I can rotate this torsion, rotate this like so using the left mouse. Just going to undo, go back to the original. So that's rotating torsions. If you want to delete an atom, you can click here and just click on the atoms you don't want. It will delete those. Evaluate the score. We got higher again if we lost the hydrogen bond. So now we can build it back just by redoing the original, or undoing what we did. So now we have the hydrogen bond again. Okay. So we may want to explore some other functional groups. So in order to start building, it's a good idea to display what we call that ligand pocket surface. That gives you an idea of where you can start building into. So for example, if I try and add a group at the back of the pocket here, it's going to try and fit it in. I click on the group that I want, and then I click on the hydrogen, and you might see it hopefully through GoToMeeting. You can see. So that was a terrible modification. We have massive clashes with the protein, which is highlighted by these red spheres. So we undo that modification that's at the back. So maybe we'll have more luck in a region where we have some gap. So if we rotate the molecule a little bit, we can go to meeting lags a lot compared to what I'm seeing and what you're seeing. But so now you should see that we have a gap here we could play around with some modifications. So let me just orientate it correctly. So we have the hinge here. Okay. So you could try maybe a halogen at this point here. See if that, and just make the change, evaluate the score. The score actually improved slightly with this halogen here. So we may be onto a good thing with these halogens. Maybe we could try a selection of halogens at this point. So we could select fluorine, chlorine, bromine, iodine, and then go to our hydrogen atom here. And click on the hydrogen. And that will run a small virtual screen at that point, at this point here, of the groups that we selected. Then we get a table here ranked by VLS score. Now you can see there's two scores. This one is if you don't have the VLS module, you'll get a score and the strain. The difference between the VLS score and the score is actually the strain. So the VLS score takes into account the energy of the ligand outside the pocket as well as inside the pocket. If you have the VLS score, you may as well just use that for ranking. So we ranked it. So this table shows that the bromine, the bromine is an improvement here. If you double click, it loads this ligand. If you want to toggle just without loading it in, you can just toggle. So this iodine, you can just toggle on and off here using this box. And we keep going down. The chlorine is an improvement as well. And actually all four of them are an improvement on the original ligand, which is the hydrogen at that point. So if you want to try additional groups, or if you want to try your own, you can add your own group here. So you just click on Add New, and you need to sketch the ligand. So you can right click on the atom and choose attachment. So you can see each of these groups have an attachment point with it's an asterisk. It might be hard to see if you go to meeting, but so you right click and you say the attachment point on the atom is here, and then you can just add a group. And then close it. It'll ask you to save changes. You go yes, and then you have your new group here that you can try. So you just click on that, and you can use that. So they had a question about the score and the VLS score. So yeah, it's quite confusing. I would stick with VLS score. The VLS score takes into account the energy of the ligand outside of the pocket compared to inside. So it's the strain. That's the difference between the two scores. So we have the score and we have the ligand. So we can edit in 3D. So to edit in 2D, we click on this button here. This opens the 2D panel. You can make it a little bit bigger. I'm just going to close this for it. So here we have this property monitor, which you can toggle on and off. It's going to center the thing. So as you make changes, you can see the effect of those changes on the properties such as Lipinski's rules, what have you, solubility, log P, polar surface area, and also flags the groups as well. So if we want to carry on exploring this region, we can change this maybe to, just going to close this panel. You can see that the properties are changes on the fly. It's just a typical editor, so you can use the erase button here if you make a mistake. You can use the keyboard for shortcuts to certain atoms. And we have templates inbuilt here, for example. Okay, so we've sketched it. So then to commit this change to 3D, we just click on here, and it adds the group we added, and we can see the effect on the score. So the score didn't improve very much with this sulfonamide. Wanted to just change it to a sulfonyl maybe, see what happens. Score goes a bit higher. But yeah, so that's basically the 3D and 2D editing capabilities. So there's also inbuilt into this chemical dictionary, so you can type in a drug and it will sketch it automatically for you. So if you did that, then it would dock this molecule into this pocket as it is. Okay. So obviously we're not interested in this one, but we can undo again and go back to our original molecule. So just quickly go back to the PowerPoint. So when you make changes, it's a good idea to keep them and to save them. So one way to do that is just to, as you make them, you can save them to a chemical spreadsheet. The chemical spreadsheets are pretty interactive. You can build plots and a variety of other things, chemical searching, editing, clustering, and such like. You can save your modifications in PDB format to a chemical spreadsheet, as just mentioned, or to STF, and export to Excel or HTML. And you can also, if you make a modification and you want to share it with a colleague, you can save it. I'll show you in a minute, and then you can load it in the free iMol view, which is on iPhone, iPad, and Android devices, or in the free ICM browser. And your colleague will see exactly what you save. They don't need any special licensing or anything. It's all free. So if you're a chemist and you want to share it with a actual chemist in the lab, you can save your designs and show them in a fully interactive way. So with this example, so back to the original. So if I make, here I have the bromine modification. If I like this modification, I go to Lig Edit, and there is a button here called Add Current Ligand to Table. This makes a new table which contains the 3D coordinates of that ligand. So you can just so if we make if we make a modification to this, we can always go back to our original ligand just by double-clicking on this table. It takes us back to that design. So it's worth keeping. If you have something you like, you just save it, and you can always go back to it. If you click and hold on here, you can also tag certain things. So you can tag and give it a name, on this series 1 or something from Ring, and just go OK. And then you would get another column with your tag. So you can start clustering them into groups of modifications on certain points in the ligand. If you wanted to save it as an ICM object, if you want to save your receptor and your ligand in one object as a PDB file, you can just drag. I'm not going to do it because we're going to carry on with this, but you can just drag this into the receptor, and then right-click on the receptor and choose Save As, and then choose PDB. It will save it as a PDB file. Likewise with the chemical spreadsheet, if you want to export it to Excel, you right-click on the tab, choose Export to Excel, or Save As STF, for example. Then, so we have we have that's how to save the modifications. In terms of 3D modifications, there's some options in the Advanced tab, which I will point out. Okay, one of them being Find Group. So basically, this will do a virtual. If you click on the Advanced tab, and you click on the atom where you want to screen to, this will find the best replacement group from. We have a database of these fragments, and it will try each one on each point and give you a score. I'm just going to show you the results of that. So it's called Group Scan, and so you'll get a table in a similar way. You can rank it by score here. You can maybe build a. So in this example, we have quite a few. I'm just going to undisplay. I was going to display it in preview again. So if you double click, it loads that ligand in. You can find where it's. Yeah, so the changes are hit here. You should see it in a second. So GoToMeeting is very slow today. So so you have the sulfonamide sulfonyl here, and this one you just double click, and as before, you can just toggle on and off. You could maybe build a plot of VLS score against log P, and have different coloring for different things, so you can go through the table and look for those substituents which might be more interesting to you in terms of the overall log P. Some of these are out of over 5. You may want to discard those, or I don't know, but you have some parameters here that you can use to filter this table. Hit this. So we have an inbuilt table that you can screen, or you can also add your own substituents using this option, or add directly to the table that we use for screening. So that option is in the Advanced Find Group. The Find Linker, you need to go back to last week's webinar on fragment docking and linking, and that we used that there. And there's also a multiple position group scan, so you can click on here and you can try groups on each position in the ligand on the hydrogen, and it will rank which by score as well. And doc table, you can read in an SDF file and just click here, and you can dock a table of compounds to your pocket, which I think we showed in one of the previous webinars. Define covalent ligand. If you've got a covalent binder, you need to use this. And you can watch the YouTube video. And this also was included in the flexible receptor docking tutorial. So you can define certain groups or certain residues that you want to be flexible during docking. So for example, in this example, with this lysine might be flexible, it might come in and out of the pocket. So you can select that lysine like this using the residue selection tool. Click on the residue, and then go to Advanced and choose Define Receptor Flexible Groups, and you can say I want this full side chain to be flexible during docking, and go OK. Or you can choose certain hydroxyls to be flexible on the water molecules as well. So now we're moving on to the docking side of things. Inbuilt into ICM, into the ligand editor, you can dock at the modified ligand. You can dock, as I mentioned, from a chemical spreadsheet or table, because when you read in the SDF file, it becomes a table. You can dock to multiple conformations of the receptor. You can refine the side chains in certain situations, and you can also do ligand-based docking, which I'll show next week using atomic property fields, covalent docking, fragment docking, and docking using tethers and distance restraints. So we will do that here, finally. So in this example, it's probably best when you're making modifications to keep this ring in place because it's making these key hydrogen bonds with the hinge, which is quite typical. So if we want to keep these atoms, I was going to undisplay, or ribbon, sorry. So if you wanted to keep this ring, these atoms here in place when we dock, we select Tethers, this button here, and then click on the atoms that you want to keep rigid during docking, or keep in place during docking. So you can see, hopefully, eventually you go to meeting, we'll show you, yeah, the red crosses through the atoms that are going to be kept in place. And you can see in the table below, you have the option to use them, or at the moment they're all checked, so they're all going to be used. You can turn them on and off and display them. You can also set distance restraints. So the button above the tether one is for distance restraints. So you click on here, you click on one atom of the receptor and one atom of the ligand. Click on distance restraint. Like that. So now you can see we have 4 position restraints, which are tethers, and 1 distance restraint. And you can play around with the maximum distance by editing in the table here, and the minimum distance. Or just delete ones you don't want to use. So now if we dock, so once you've made your modifications, some small modifications just require minimization, but larger ones you may want to fully redock. So we click on the docking, redock, ligand and current receptor button here. And then it docks the ligand. So it takes a little bit of time to run the docking, but you'll get a hit list of the top conformations. So now you see that we have a table now called doc confs. They're ranked by a BLS score, the lower the better. So if you double click it, basically reproduced the crystal structure pretty well. If you want to see other conformations of the ligand, you can click on the toggle button here, and you can see the other conformations docked. So if you want to load this one in to be the ligand that you want to modify, you just double click and it will load it into the ligand editor. You got the score. Okay, so if you had multiple receptors, if you go back to the video from a couple of weeks ago, you can click and hold and it will dock to a stack of receptors as well. Flexibility. And you can also, once you've docked, you can also refine the side chains. There's the option to have side chains being fully flexible during docking, if you carefully select 3 or 4 of them. But you can also just refine the side chains by clicking on Advanced and going here. Thank you very much for attending and your support of the webinars. We appreciate it. Next week's webinar is on the atomic property fields.